Okay, so now we're going to finally move on to the genetics part of the unit, and this follows chapter 11. Right now we're going to focus on Mendel and his P's and what is considered Mendelian genetics. So Gregor Mendel himself was a monk. I feel like all of you must have learned this in freshman biology, but basically he worked in the garden and studied pea plants. And he used peas as his model because he could control mating. So as plants, you can either self-pollinate, meaning that you take the male and female parts from the same plant and use that, or you can cross-pollinate, meaning you can take parts, they do sexual reproduction, as we learned in the last PowerPoint, to cross-pollinate and create many varieties. And there are many different traits that he could look at, and they're quick and fast to grow. So here's oftentimes what he would do. So he would take the P generation. So as we go through, we're going to kind of have to remember a lot of the vocab that you learned once upon a time in the abbreviations. P stands for parental generation, and they have purple flowers and white flowers. These are called true breeding, meaning that we will learn that they're homozygous, but every purple flower makes purple flowers, and every white flower is going to make white flowers. They have been self-pollinated, meaning that they only ever fertilize themselves. So therefore, they only ever create kind of genetically identical versions of themselves. So if you take a purple one and a white one and you cross it together, they have the first filial generation, or the first generation to come after the parents. And he found out, Mendel did, that all of the plants had purple flowers. And these are called hybrids because you have mixed two different genetic parents together. So that's why it's called a hybrid, even though all the plants were purple. And then he allowed these plants to self-cross or cross-pollinate because at this point they're all really genetically identical. And then he noticed that in the second generation, or the second filial generation, that there was kind of this 3-to-1 ratio of purple flowers to white flowers. And that got him thinking of how this could possibly work. Okay. So generally, this kind of goes through the vocab of what I had mentioned. All right, so alleles. We've learned what a gene is. A gene is a segment of DNA that codes for a protein. Alleles are really alternate versions of that gene, and they can contribute to things like hair color, eye color, ability to taste bitter, in this case, whether a flower is purple or white. So on a pair of homologous chromosomes, let's say this is on chromosome 1, don't really know for sure, but one of these would be mom's chromosome 1, one would be dad's chromosome 1. And in the same location on each one of those chromosomes is the gene for flower color. And there are alternate forms of that gene. So you could have the allele that will confer purple flowers, or you could have the allele for white flowers. So it doesn't necessarily mean that it's like disease or not disease. It's just the particular version that you happen to have. So Mendel was able to look at 7 specific characteristics in pea plants because he was able to see many different things. And so he started crossing them together and noticed this interesting 3-to-1 ratio in offspring across many different traits. So he arrived at the words dominant and recessive. Dominant is the trait that's going to be expressed, or the allele that's expressed when present, whereas the recessive is the one that gets hidden in the presence of a dominant allele. And so based on the specific ratios he was able to get, and the fact that the F1 generation had certain traits that were completely hidden Like purple and white flowers together only make purple flowers while the white was hidden. So therefore it must be recessive, led to that vocabulary. Okay, so he came up with a specific set of principles. Alternate versions of genes, so alleles like purple flowers or white flowers, they can cause variations in traits. That makes sense, right? We all have different hair colors and eye colors and so on. For each character, or I guess in this case it would be really like trait, every organism inherits one allele from each parent. So everybody's got 2 chromosome 1s, 2 chromosome 2s, 1 from each parent, and there is a particular version or allele of a gene on each one of those. So for any trait that we have, we get 1 version from mom and 1 version from dad. If they are different, so if the alleles are different, the dominant one is the one that we see. The recessive one gets masked. And then I guess as an extension of that, if there is no dominant one, then you will actually see the recessive. So he arrived at something called the law of segregation. For 2 alleles, the 2 alleles for each character separate during gamete formation and is passed to the offspring from parents. So if you think about meiosis and what happens, the chromosomes separate randomly, and those can get passed from parent to offspring. Okay, so here's an example. Purple flowers, if they are true breeding, you can only give big P's. Dominant traits get capital letters. Recessive traits get lowercase letters. So a purple flower in this parental generation can really only ever create sex cells or gametes that have a capital P to give. White flowers can only give a little p. So when you put it together, all of the offspring are big P, little p. That's where the hybrid comes in. This is what we will learn as heterozygous. Hetero means different. Well, half of the genetic material can be capital P, and half of the genetic material can be little p. So during meiosis, when those sex cells are created, there's kind of a 50-50 chance that it'll be this. So when you cross them together, you create this magic guy right here. This is called a Punnett square. And it's a representation of how different combinations of parents can produce offspring. So, for example, one parent could give either big P or little p. The other parent can give either big P or little p. And then depending on how those fertilize or come together to make an offspring, you get different resulting genotypes or genetic makeups. It could be capital P, or capital P, little p. All of those instances are going to result in purple flowers because it has a capital letter, which is dominant. Little p is going to result in white because there is no dominant letter to mask that trait. But the idea is it can be random. So anytime these 2 plants come together, it kind of gives you a % chance of having different phenotypes or physical appearances. Okay, so here's kind of a refresher on some of those vocabulary terms. Again, dominant gets a capital letter, recessive gets a little letter. Homozygous, homo means same, so it's either going to have 2 capitals or 2 lowercase. Heterozygous means different, so it's going to have 2 different alleles. This is where there's a capital and a lowercase, and depending on the genetic combination, it's going to yield a specific phenotype. Phenotype is physical, in this case purple or white. Genotype is genetics. It's important to note that 2 different genotypes here can lead to the same phenotype. So the ratios are not always the same. Phenotype physical, genotype genetic. Okay, so I'm returning to this Punnett square business. This slide just kind of introduces you to it, but the idea is you set it up in something like this. And we'll go through many examples, but this is one that is called a TESTCROSS. And a TESTCROSS is really if you have a dominant phenotype, let's say purple flowers, but you really don't know its genotype. You're unsure if it's homozygous dominant, like big P, or heterozygous, big P, little p, because both of them make purple flowers, and you're just unsure. So they perform what's called a TESTCROSS, and this will confirm the genotypes of that parent. So what you do in a TESTCROSS is you take your unknown and you cross it times the recessive phenotype. In this case, little p are white flowers, because you automatically know what that has to be. Anytime you see the recessive phenotype, you know that they had to have 2 little letters to get there. So there are predictions you can make. Well, on the left here, if the purple flower is homozygous dominant, then you would expect to see 100% of all of the offspring are going to be purple. But if the parent is heterozygous, well, then you'd expect to see about 50-50. Half of them are purple or half of them are white. So after this cross, basically, if you see white flowers, you know the parent had to be heterozygous. And if you don't see any white flowers, well, that indicates that the parent is homozygous dominant. Okay, so there's something called the law of independent assortment. This is something that Mendel came up with as well. He said that each pair of alleles, so each kind of versions of traits, segregate or separate independently during gamete formation. So there are alleles for P color and there are alleles for P shape. Now, when they are, splitting up into the gametes, it's random. They do this independently of one another. We will learn about instances later of linked genes, but the assumption here of law of independent assortment is that different alleles are independent of one another. So, for example, here, in this particular experiment, they looked at shape and color. So yellow is dominant to green, designated with a capital lowercase y. Round is dominant to wrinkled, so that's capital R and little r. Because these are homozygous, these parents can only ever make big Y, big R, or little y, little r, which means that all of the offspring are heterozygous. Big Y, little y, big R, little r. Okay, well, that's when we get into this crazy big Punnett square here. So he set up an experiment to say, okay, well, if they are linked, this is what you would expect. And if they aren't linked, this is what you would expect. So what he did and what became true is the idea that you come up with all the possible combinations of alleles. So you could have big R, big Y, big R. You could have a big Y and a little r. You could have a little y and a big R, a little y, little r. Same thing for the other parent. So there's really 4 different combinations that you could make. It's kind of like foiling in math class. And then based on that, you come up with all of your possible combinations. And then that yields something called a 9-3-3-1 ratio, meaning 9 of 16 are going to be round and yellow, 3 of 16 are going to be round and green, 3 of 16 are going to be wrinkled and yellow, and only 1 is going to be wrinkled and green. And this is what he observed. If they were linked, you would expect to see something a little bit different. And so based on what he saw, he was able to arrive at this independent assortment idea. Going back to what I was mentioning, a monohybrid cross is 1 trait, 1 character that you're looking at, color. Di means 2, so a dihybrid cross, you're looking at 2 things, which is that bigger Punnett square that's really 4 by 4 instead of 2 by 2, like this. This one's a little weird because it's kind of in a diamond formation, but you get the point. Okay, so here's an example of a dihybrid cross. Let's say I wanted to put these two together. We can work an example out, but you would come up with all of the possible combinations. So you could have a big A, big B, big A, little b, big B, and little b, little b. So you would line those four possibilities up along the top, and since the other parent is the same, you would line those possibilities along the side and then fill it in. Okay, so probability here. Now we're switching gears a little bit because probability really governs Mendelian genetics. So if you want two events to occur together, so let's say you want a yellow and round, something like that, then you end up multiplying their probabilities together. Or we always use dice as an example. So if I have two dice and I roll 2 6s, what is their probability of getting each one of those dice to roll a 6? When you want two events to happen, you multiply their probabilities. So on the first dice, if I roll a 6, I have a 1 in 6 chance because there's 6 total sides on a die. One of them has a 6 on it. So I have a 1 in 6 chance. On the second die, I have a 1 in 6 chance of rolling a 6. Now, if I want both of them to be 6 at the same time, I multiply their probabilities. So altogether, if I roll 2 dice, I have a 1 in 36 chance of getting 2 6s. All right, probability of having 5 boys in a row. Well, every time you have a child, it's a 50-50 chance. So you have a half of a chance. And regardless of how many children came before, every time you go to have a child, it's always a 50-50 shot. So to have 5 boys in a row, it's a 1 in 32 chance because you'll take 1/2 5 times. Okay, so now here is a little bit tricky. If I have a crazy trait thing here, so I have A's, B's, and C's, what is the probability of having them be big A, big B, little c, little c? Well, you multiply their probabilities together. So let's look at just the A traits. In order to get big A, little a, if I were to do a Punnett square with just the A's, I would have a 1/2 chance of getting big A, little a. Same thing with the B's. If I did big B, little b with a Punnett square of big B, little b, half of them would be big B, little b. And then last, I'd have a 1/4 chance of getting little c, little c. So if I multiply their probabilities together, I would get 1/16. Okay, now the rule of addition is a little bit weird. Probability that two events will occur, but they don't necessarily have to happen together at the same time, or order doesn't really matter. So in this case, let's say I want to throw a die that will land on a 4 or a 5. So either one is acceptable. And so that's kind of two options. So I have a 1 in 6 chance of landing a 4 and a 1 in 6 chance of landing a 5. So I would add their probabilities together. So if I roll 1 to land a 4 or a 5, I have a 1/3 chance. Okay, so let's say segregation of alleles. If I have a parent who is big R, little r, you have a 50-50 chance of getting an egg with a big R and 50-50 chance of getting an egg with a little r. Same thing for the sperm. And then that's why you end up with this combination of events. So that's why we are really able to use Punnett squares, and we multiply their probabilities together. Okay, so now we're going to start to get into the weird situations where Mendel didn't quite understand what was happening, or P's didn't really give a good representation that we're going to learn about later. So complete dominance is what he observed. It is either the dominant trait is observed, and it's indistinguishable whether it's homozygous dominant or heterozygous. So if you're big Y, or big Y, little y, either way, bright yellow seeds. That's complete dominance. Now, there is such a thing as incomplete dominance. So in certain instances where you would have like a red flower and a white flower, sometimes the offspring is going to be intermediate between the two parents. So you end up with a pink flower. So red is dominant, but it's not completely dominant because otherwise you'd have red flowers. Instead, you have pink. So here would be an example of what that looks like. So in the instance of that, you would end up with plants that have red, pink, or white. Now, also in here, it's important to know that for incomplete dominance, and we'll get to it later too, you don't just put the R's or the W's. You could, but they like to put them as little superscripts. So it's kind of like color red, color white, color white. And that way it shows that if you have two together like that, that you'd end up being pink. It just avoids having two letters, so to speak, in the Punnett square. Other things that are included are codominance. So codominance is when both of them are expressed together, which we will learn about. Red hair and white hair coming together to make a roan horse. Roan doesn't necessarily mean like in between red and white and making a pink horse. That's not true. It's when you look closely at a horse in that instance, they have individual red hairs and individual white hairs. Whereas in multiple alleles, now we're getting into something a little bit different. Multiple alleles means there's more than two options, and that does happen in some traits. So blood groups are the perfect example of that. You can see there's A as an option, B as an option, or those are both codominant, meaning that if it's there, it will be expressed. So they are equally worthy. And then there's a little I, and that's the recessive. That's what we refer to as O. And so some people like to write their genotypes like this right here, as IA, which means you'd have type A blood. If you have I, capital A, little I, you would also have type A blood because the little I is recessive. Type B blood can be either big B, or big B, little I, or nothing. Some people like to draw it as AA or AO, BB or BO. I know I do that a lot. If you have type AB blood, you must have one of each. And then in order to have type O blood, that is the recessive, you have to have little i's only. And this has to do with what carbohydrates are on the surface of, or like what antigens are on the surface of a blood cell. And it helps designate what type it is. Okay, so here's just the comparison of the phenotype on the left, so what the actual physical appearance of that would be, and then the genotypes on the right. Again, this is designated with I's. Some people like to write just AA or AO in place of the I's. That works too. Either one. All right, so this chart just gives you a picture of who can give blood to who. So O negative is considered the universal donor because there are no specific surface antigens that your body could reject. So they can give blood to anyone, whereas O negative can only get blood from O negative because it will have a poor reaction with anybody else. AB is considered the universal recipient because it has antigens to everything. It can actually receive blood from anyone. The idea here is that your body will accept blood that has the same antigens as yours. It will reject or fight against any antigens that are present that are not yours. So if you have B blood, you can't get anything that has an A. If you have A blood, you can't get anything that has a B. If you have type O blood, you can't get anything that has an A or a B. And then uniquely, if you're an AB, well, then you can kind of get everything. The additional piece of a plus or minus comes from something called an RH factor. It's an extra protein. And if you're positive, you have it. And if you're negative, you're not. It's kind of like a additional antigen. So negatives can't get the positive, or there might be a negative reaction. I'm going to have you work out that problem piece on your own. The next slide goes into something new and different, polygenic inheritance. So poly means many, so many gene inheritance. So 2 genes acting on a single character. So in freshman biology, we lie to you a little bit and say that, there's a singular trait that confers hair color or eye color or whatever it may be. But really, it's not just one. There are certain phenotypic characters like our height or our skin color that really are controlled by multiple genes. So depending on the exact combination of all of those genes together, in each one of those versions, it can confer different things, like our skin color. So, for example, I'll take my own self. I must have gotten whatever the palest possible combination of gametes you can get, because I am like the palest white person that exists. So my phenotype is going to be over here in the 1/64th. But depending on what combination of all of those genes someone else got, it can mean darker and darker and darker skinned. Another thing to think about is nature versus nurture. It's kind of weird to think about, but the environment can also influence the phenotype. So your genotype is not the sole determiner of your phenotype. Genetics does play a role, obviously, but there are environmental factors that can influence the way that you look. So for example, these are a picture of hydrangeas, and obviously they have a genetic code conferring their color, but depending on the acidity in the soil and the aluminum content that they're grown in, that influences the exact intensity and shade that those flowers actually become. Okay, so this is also where we introduce the idea of something called a pedigree or a family tree. It shows the relationship between parents and offspring. So you got to be able to look at one of these and know how it works, who's affected by a disease or has the whatever trait that we're talking about. So women are usually circles, men are usually squares. A line between a male and a female means that they came together to generate some kind of offspring. And then the line down shows those offspring. So in this particular example, George and Arlene had 4 children. They had 2 girls, Wilma and Ann, and 2 boys, Tom and Sam. And it looks like Tom got together over here with Sandra and had 2 children, Daniel and Alan. Alan got together with Tina and had a child. So this is really conferring 1 generation, 2 generations, 3 generations, and even 4 generations here. The ones that are filled in are expressing whatever trait. So Arlene, Tom, Wilma, and Carla all happen to have whatever trait might be discussed here. So when we're doing a pedigree analysis, we can really look at certain traits. So one of them is a widow's peak. Another one could be whether you have a free or attached earlobe. So on the left, we have, is a widow's peak dominant or recessive? Well, based on this, it is actually a dominant trait. Because you can see that a person with this genotype has it. All of the ones that are all lowercase don't. So if you have a capital W or a capital little w, then you have a widow's peak. And again, if you don't, you don't. Whereas earlobes, if you have little f, then you have an attached earlobe. And only those that have little f have an attached earlobe. So this is what an attached one looks like. You can see a free earlobe kind of has that extra piece out here. Attached is recessive and free is dominant. Okay, so genetic disorder examples. Recessive disorders, you might have heard of before. So you would need 2 alleles of this, 2 little letters, homozygous recessive, and that's cystic fibrosis, Tay-Sachs, sickle cell, phenylketonuria is another one. Whereas autosomal dominant, meaning if you have one capital letter, you don't even necessarily need 2, you will get the disease. You will have Huntington's, and it's called a lethal dominant. If you have 2 capital letters, it's considered lethal. Okay, so this particular table goes through a lot of the different examples of what we discussed. So typical Mendel and his P's is complete dominance. Red and white making pink is incomplete dominance. Codominant would be an example of roan cows or AB blood groups. Multiple alleles is also blood groups because there's more than 2 options. And one word that we didn't really talk about is pleiotropy. What that means is that there is one particular gene that can have a lot of phenotype characteristics. So one gene can affect in some way many physical traits. So sickle cell is one example where that one gene has the ability to control multiple physical things. Another word that we didn't quite cover is called epistasis, where this is really the phenotype expression of one gene affects that of another. So it could have to do with these instances of these 2 genes, the B and the E gene, affecting each other in the sense of coat color. And then polygenic, single physical trait affected by multiple genes. So skin color was kind of our example of that. Okay, and then at the end was really the beginning part of your PowerPoint. There are a couple of sample questions that you can go through to kind of practice some of those instances of genetics.